First of all, welcome everyone to this webinar. We're so excited at the turnout that we have, and we're just very excited to be talking with all of you today. Before I start, just if you take a look, we have the schedule for the upcoming webinars. We have two more happening, one on October 12th, which is really talking about how to manage big data and handle it when you're in both analysis and collection. And then November 3rd is talking about how to handle the new NIH policy that's going to require data sharing for all grants. And we have some really great resources and ideas for that and how the NSRR can help. So just a little bit of housekeeping. Please note that we'll have all questions answered in the Q&A for each speaker. After each talk, I'll be fielding some questions from the written questions. And we also have a poll available if you're able to see, just to learn a little bit about where everyone's coming from. So with that, we're just so excited to begin today. What the goal of this overall webinar is, and this has been a wonderful collaboration between NHLBI and the National Sleep Research Resource. And our goal in the NSRR is to make the sleep community aware of really the best ways to utilize all the resources that we'll talk about in ways to support sleep and circadian research. But part of that is we want to interface with the community to learn about how we can enhance the NSRR. So that's why we're looking for great feedback from all of you attending today. But in general, the goal of the NSRR is to really promote sleep research by encouraging further growth of sort of big data resources like the ones that we host, facilitate best practices in both analysis, methodology, ideas of signal processing, secondary analysis, and also in sleep data management. And so with all of that in mind, we've presented this seminar series sort of describing all the many ways that, that the NSRR and community can and have helped support each other and support advancement of sleep and circadian research. And my name is Michael Prerau. I'm a researcher who and faculty and part of the outreach portion of the National Sleep Research Resource. And so I'm happy to help facilitate today's talk and questions. So without further ado, we'd like to start the first talk. And this is from Susan Redline, who I have the great pleasure of introducing today. She is the Peter C. Farrell Professor of Sleep Medicine at Harvard Medical School and the Director of Sleep Medicine Epidemiology at Brigham and Women's. And she is a founding director of the NSRR and is co-PI along with Dr. Sean Purcell. And very excited to have her speak today. Susan, please take it away.
Just see, sorry about this. Give me 1 second, two things were open here. Okay, good. Okay, well, thank you. Can you see the slides? Okay. Yeah, okay. Well, again, thank you all for joining us today. I'm really super excited to have the opportunity to provide an introduction to the NSRR. And I'm also just delighted that we have some contributors to the NSRR who will follow my talk, as well as a couple of users. And I think that they will really beautifully exemplify really the power of building community, contributing data, developing new collaborations. But my goal right now is to really kick off the meeting. And what I'm going to do is I'm going to address the question of related to why, what, and where to. And the what question is, what are the gaps that NSRR is really aiming to fill? So I'll briefly talk about the potential for big sleep data and open source analytics to really promote our field. Number two, I'll answer and that's the why. And then the what is about NSRR, who funds us, what our mission is, a little overview about our content and structure, and specifically, what is the value that we might bring to data that's deposited in our repository. And I'll conclude with some of our next steps, really highlighting integration with Biodata Catalyst. And as Michael just told us, I'll just mention our data management sharing plan. And please note that many of the things I'm going to talk about today, I will be talking about very quickly, but they will be gone over in more detail in subsequent webinars. So I'd like to really begin by highlighting that the sleep field is really uniquely positioned to take advantage of big data. In fact, our discipline is organized around diagnostic tests that collect hours and hours of data from multiple channels, really providing rich sources of respiratory, cardiovascular, brain, and movement data. And in fact, the potential power of these data in an era where we have growing availability of advanced analytic tools has been recognized by our own societies. So for example, with the progress in machine learning and artificial intelligence, which nonetheless requires big data, the American Academy of Sleep Medicine has begun to advance several position papers and visions for how machine learning might be able to be used to really complement, potentially replace manual scoring, as well as to identify new features that predict health outcomes better. Similarly, in this second article that I was an editorial that Dr. Purcell and I wrote regarding a big data in this journal Sleep from last year, we discussed the opportunities to further harness data and technology, not only just that focus on the sleep data, but then integrate multiple streams of data relevant to sleep. So it includes sleep, but as well as upstream factors such as genetics and environmental exposures and behaviors that influence sleep, as well as downstream types of data, data that may help us understand the impact of sleep on gene expression, methylation, and clinical and physiologic outcomes. Now, this excitement, though, is tempered by really the traditional concerns I know all of you have shared, at least I have, is that it has been over the years hard to access well-annotated sleep data, as well as sleep data that are linked to these other streams of data. And the problems are ones we well recognize. Data, whether it's clinical or research, tend to be stored and owned in silos. They've often collected, although in a rigorous way, they are often collected in ways that are poorly standardized that prevent an outside user to really maximize their utility and combine with other sources of data. So there might be different collection protocols. There's lack of standardized montages across our sleep labs, variable scoring, variable use of labels. Our equipment manufacturers, and I think some of you are out there, I call upon you to work with the sleep community to get rid of unwanted filters and inconsistent exports that impact our data. And our own sleep community as well, there's a need to more consistent use of the terminology, the labels, the metadata, as well as corresponding sort of standardized common data elements. And these, and over time, even the data that have been made available in the public, for example, in BioLink and other data repositories, have either been simple summary sleep data without that rich signals, or if they had the rich signals, they often didn't have rich clinical outcomes. And that really was the motivation of our team to create a concerted effort to both curate and aggregate data relevant to the sleep community. So our, in brief, our history goes back to a little before 2014, when, with wonderful support initially by Michael Turi and now by Mariska Brown, we got a resource grant to initiate and maintain a repository for sharing large amounts of sleep data, of course, initially from multiple research cohorts and clinical trials, but we're also given the green light to expand these to multiple other sources of data, including clinical data and data generated, even experimentally and even in animal data. And in 2019, the NHLBI converted our grant to a contract. So we are really working on behalf of the NIH or NHLBI particularly. And our mission is here. We really want to work with you, the community, to help advance sleep and circadian science. And that would be through our repository that would support secondary data analysis, algorithmic development, and advanced signal processing. And the actions are sharing high quality data and open source tools. Exist out there. Not only do we need these large data to use genetics, the association of sleep to hard outcomes, by that I mean death and cardiovascular events, promote precision medicine, look at sleep across an entire lifespan, as well as have better algorithms. And all of these needs require large samples, standardized or harmonized phenotypes. They need diverse samples that do two things. One, allow us to break down the heterogeneity in the population and our disease, as well as to ensure that we are making inferences on really diverse populations and avoid really further disparities in our research and clinical work. And also for the rigor of research, we want to make more data available so people can have independent samples to replicate. And as I mentioned too, we need large samples to really support these advanced algorithmic work out there. So what does NSRR consist of? There are really three main areas that we'll talk about. One is our data set, simply our repository, which really focuses on polysomnography and actigraphy signals, as well as the summary data, as well as sleep-specific questionnaires collected by researchers and clinicians, as well as sets of standardized or harmonized metadata and terms, and in fact, linkages to other data sets, for example, in dbGaP, which contains genetics data. We also, and this is much of the activity of my collaborator, develop open source tools for EEG, and some of making their tools for respiratory endotyping available as well. About that as well. And briefly, our repository, as many of you have gone to it, really provides you with the ability to browse, search, download the sleep research data. And over time, we're hoping to make it easier and easier for you to identify harmonized terms, core variables, across common data sets, and really promote really best practices. So here's a brief overview again of the data contents. And I just wanted to note for those of you, who are less familiar with NSRR, the scope of data per cohort is quite variable. So currently, I think we have about 18 different data sets that we're actively making available. Most of these are research data sets. They were observational studies. Some of them were clinical trials. Although there are a few other sort of oddities. We have one individual that donated a year of his own actigraphy data, and that data, for example, lives there. But each of these data sets are very different in how they were collected and the governance that's attached to them. So whenever possible, we try to share as much of the data that could be linked to the sleep data, from demographics to blood biomarkers and cognitive testing. But in some cases, because of governance issues, the data other than the sleep data are a bit more limited. And here is, for example, an at-a-glance spreadsheet that you can access through NSRR. And this really lists the sort of the snapshot of the key data sets available using their initials down the columns, and then down the rows, and then across the columns, you could see broad categories of types of data that you might find in each cohort. So this may actually give you a snapshot view of what cohorts, for example, have neurobehavioral outcomes, and you could just go down that list. Now, what have we actually made available and shared? So currently we have about 8 terabytes of data. We have actually approved almost 4,000 data use agreements from researchers around the world. And those researchers include everyone from high school students through early stage investigators, senior investigators, and even individuals from industry. And we've had, over 900 terabytes of data downloaded. About 2 to 3 terabytes of data are downloaded per week. And the data that you currently can access are almost 50,000 polysomnograms in EDF format, about 20,000 sleep studies that have had derived sort of quantitative signal analysis of the ECG or EEG already done and made available, over 4,000 actigraphy files, and plus a wealth of data on the various variables of terms that we've annotated. We've actually annotated over 5,000 terms, including sort of metadata that gives the sort of the background or the provenance of those terms. But it's really key that we're not just a repository, because if we're just a repository, there are many, many repositories. So we really want to not, also drive discovery and support research. In particular, we want to add value and stimulate really, interactions across the community. And we do that by taking sort of the data that comes to us and review it, and the extent possible, work with the contributor to improve the documentation, provide search capabilities to make it easier to actually find the documentation as well. We also, and this will be a subject of a later webinar, we work at identifying core terms and signals we think will be generally of interest to the sleep community. And when possible, we harmonize it. And in other cases, we standardize it so you understand what you're using. We also create metadata for most of our variables, and we follow these principles of fair and trust, which I'll talk about later. We also, as I mentioned, are in parallel develop and sharing open analysis tools, which also include exemplar applications of NSRR data. We also have the ability to link NSRR data to other resources. And then finally, through our outreach, education, social media, newsletters, we hope to continue to build community. And we very much want your input on how to do more of that. So here, very simply, if you've gone to NSRR, you see that in terms of adding the value, we've tried to streamline and standardize the documentation of individual data sets. This is one of the earliest studies, Sleep Heart Health Study. And you could see, you could click these different boxes and learn about the what, who, when, funding, and download, many of the everything from case report forms through polysomnography manuals of procedure. You also, we've organized our list of variables really into various folders that make intuitive sense. For example, cardiovascular outcomes in this data set, and demographics, and spectral analysis, interim follow-up. And you could actually navigate to these and then very easily find the terms and the definitions, and they exist. And as I mentioned, we want to be compliant to the evolving standards that relate to fear and trust. So fear here means findable, accessible, interoperable, and reusable. And these are often, you require that there's rich metadata that really uniquely describe each of the variables, and that you can, you can openly identify things. There are standardized vocabularies and so forth. And the trust principles further, talk about transparency, responsibility, a user-focused sustainability and technology. And this really refers more generally to our infrastructure and repository services. And we're very committed to that as well. Now I'm just going to briefly sort of just mention and we'll go into these in other webinars and other dates, but, a big focus of ours is in fact to make it easier to understand these various potentially heterogeneous sources of data. So Ying Zhang in our group and Matt Kim have been leading a lot of our harmonization workflow efforts. And just to let you know that we can't do this to, the 10,000 or more variables in the repository, but we've identified really core sleep concepts, core demographic variables, core clinical outcomes that we think would be useful and are present in 2 or more data sets. And we identify them, we then map them to sort of, to sort of some common intuitive labels. We look for heterogeneity and how they were combined, and then we really build harmonized terms, and we do this in an iterative process. So you may find variables, age, for example, in the data set, but you'll also may find something called NSRR age. And the NSRR age might be the term that was used that is most similar in units, or how the question was asked or derived, for example, across data sets. And here is just an example of how we approach sort of, again, this issue of trying to make it easy to find variables across data sets. Many of us are interested, for example, in the symptom of sleepiness, and often that's been assessed in many of our studies with the Epworth Sleepiness Scale. However, each data set that we got may have used different labels for the Epworth Sleepiness Score. So we've actually created tags for the Epworth, and then created really a common sort of tag across the systems. And, you could just click this variable for any given data set. And this is one data set. And you could look at each item, as well as the overall score, the responses, and also the frequency of the responses over, and in this case, over different longitudinal visits. Now, a major effort that you'll hear more about later, and we very much welcome your later input, is work that Dr. Purcell is doing with sleep signals harmonization. Even though our sleep data was obtained from research studies, it was astonishing to us that followed standardized protocols. It was astonishing to us how many times there was variation in how various channels were named or labeled, and how various sort of potential sources of noise got introduced into those primary data. So with Dr. Purcell's guidance, we've done really 2 things. Where one is we've really identified sort of some of those annoying aspects of that occurred during signal acquisition. For example, various polarity issues, or when signals were contaminated with line noise or artifact, or there was some, the devices resulted in inconsistent like staging or truncating. And those would be very hard for us at this point to regenerate. It would create, but we actually flag these. So at least the user is aware of that. And then we do 2 other things, and that is we identify as many of our, signals themselves to, we map them to really more standardized references when possible. We call those our harmonized EDFs. So that allows us to really check the channel names and annotation labels and so forth. And then and that would make it easier, for example, to select C3EEG across all studies, even though they may have been called C3A1 or C3M1 and that type of thing. And then the other thing we try to do is to actually identify a core canonical set of signals that we think that are really critical for most analyses. And when possible, we reference them, we resample them, and we try to create really a set as consistent as possible. Now, Dr. Purcell's also been working on open source EEG tools. The tool set is part of our NAP pipeline, or that's called the NSRR analytic pipeline, with Lunar being key software now available for the analysis of signal data. And you could download these directly through our application. And here's just an example of Lunar where you can actually do online some staging. You could look at queries and visualization for both the annotations as well as the spectral analysis, and really look at the sampling metrics. And this is continuously being updated and enhanced. But it's not only the tools, but it's the standardization of an approach. So in the center of the slide, you could see that we are working toward really pipelines that will work not only for our data now, but potentially even for data that's not even in NSRR now, where you could use open source tools and processes to, for example, do initial QC, for example, checking the polarity of signals, then doing standardized automated staging, and then doing some maybe advanced metrics with your EEG or ECG, for example, deriving like spindle dynamics, and then doing advanced respiratory analyses. So we've also been working on that, and you'll see more and more updates as time goes on. Now, finally, I just wanted to conclude with some of the community outreach. And again, this is part of what we're doing today. And both Dr. Prero and Paige Sparks, who's also going to be speaking to you, we have a blog, as you know, a forum for both support and discuss items. The webinars that were just kicked off. We hope to work with societies, and we have a presentation next week to the Sleep Research Network of the SRS to get their input on some of our harmonization and other efforts. We also try to be active on social media and generate periodic newsletters. We try to highlight, the research that's published from NSRR and also track those publications. So here's just an example of blogs where we try to highlight your work or new initiatives, and as well as any new tools launched and new data sets that are coming on board. And here's just a snapshot of our forum. And if we sort of just take even that first forum, post-insomnia subjects in Mr. or sleep and health, and the user had a question, you could see, one of the senior staff folks, Michael Rushman, who many of you probably know because he really has been fantastic with support, will actually point people to the clarify questions they have about availability or any other quirks that weren't available. And then really to conclude, I wanted to say we still have so much more work we want to do, and we want to do it with you. We are very actively working with NIH to integrate and migrate and integrate many of our data sets into NHLBI's cloud-based platform that's called Biodata Catalyst. That will enable a user to actually use cloud-based tools to do their analytics. But more importantly, many of our cohorts are also represented already in the cloud, where there might be imaging data or genetics data, and it may be even easier to kind of integrate those streams of data in that cloud platform because of both technical and regulatory issues than in NSRR. As Dr. Prero told you, and we'll talk about this in another webinar, is we are also really hoping to help the user community meet NIH's data management and sharing policy guidelines. As I mentioned, we're very interested in really getting input into how we are harmonizing and standardizing and sharing data, and really making it a bi-directional dialogue. And more cohorts and data sets are continuously being added. In the near future, you'll see a very large retrospective pediatric data set added, as well as a prospective pediatric data set of very young infants, as well as some animal data and circadian data. These are all new data types. And then, as I mentioned, we continue to work on the analytics and the new cohorts. So I just want to end by really acknowledging a huge team at the Brigham and my PI, Dr. Purcell. But for today's effort, I also want to give a big shout out to Paige and to Michael. I already shouted out NHLBI. I should also mention Pavan Gopharani, who's been involved with both stages of NSRR. And in the initial phases of NSRR, we had good collaborations from our colleagues in University of Kentucky, Beth Israel, and Case Weston. And I also want to thank them. So thank you very much.
Thank you, Dr. Redline. That's wonderful. We have two questions in the chat. The first question is, could you please expand on your plans to include animal-based data in NSRR?
That is a good question. And Michael is, Dr. Priot has actually been our first leader there. But I think the simple answer is that we'd like to create a resource that is as, it reflects the needs of the sleep community, and that's more than just human data. So, we want this is an official call out for those who have animal data to contact us if you are able and interested in sharing data. We have one data set that Dr. Priot right now is working on making available. So Dr. Priot, do you want to actually, this has been really a little bit your brainchild. So would you like to say more about that?
Sure, absolutely. I mean, I think it's really interesting to be able to view sleep on these grand epidemiological levels and then tie that in on these sort of multi-level analyses. And so for the NSRR to have human data and rodent data and, maybe primate data at some point, I think that would be a really wonderful boon. So we're reaching out and working with several of our colleagues who do this to bring those things online. So to be continued, but we look forward to opening that up shortly. So the next question, Dr. Redline, wondering if standard operating procedures might be available for new or ongoing studies so that the data are managed in a way consistent with those in the NSRR, enhancing opportunities to combine data sets from the outset. What a good question.
Thank you for that question. And in fact, that question ties in directly to sort of how we're trying to support the community in addressing the NIH data management and sharing plans. So we're actually creating a whole workflow with everything from sort of how to create your data dictionaries, how to identify, how to create metadata, how to even create how to identify formats for your PSG, actigraphy data, etc., that will make it easier to really share. So, and that's going to be a session that we'll be giving a little bit later this fall. So thank you for that question.
Great. And it was there but disappeared, but there was a brief question about, are we going to have further webinars to actually address analysis techniques and procedures for using some of these open source tools? And the answer is absolutely yes.
Yeah.
Okay.
And I also see, thank you, Dr. Hall. I didn't realize you gave us that really great question, but I'm not surprised by your wonderful question. Thank you for joining us today. And I see there is a question about Honolulu Asian American study. And I have to say that, one of our limitations is that we're limited by what the user is willing to share. And we really had hoped, I myself had been involved with the Honolulu Asian study. We have beautiful polysomnography data on a large number of very older men. However, because of various concerns by the study, that it was an NIA-based study, we still have huge restrictions. And I know that there are obstacles to getting the data owners to approve data sharing. I do what I can to reach out to the folks at Kuakini. That's probably our toughest data set to share. So I'm very sorry about that. Reach out to me and I try to do my best.
Well, thank you so much, Dr. Redline, and please stay tuned to our future webinars to hear a lot more from Dr. Redline and all of these details of great things, along with many other speakers. Wonderful.
Thank you so much, Guy.
Okay. So now we've gone from this big picture about the overall view of the NSRR, and we're really now going to hone in on some some specific data sets and the stories about what it means to share a data set, the process of that, but also sort of the fruits of that labor, how these things have turned into collaborations and new scientific findings for the people who have shared, that have really sort of borne great scientific fruit and results. So the first one we're going to discuss is really talking about sharing research data, and this is from the Wisconsin Sleep Cohort. And so we'll be hearing from Dr. Erika Hagen and Matthew Gratton. And they're both going to sort of talk about first the overall scope of the data set, and then we'll talk about the application. So Dr. Hagen is an epidemiologist and a senior scientist at the Department of Population and Health Science. And she helps to coordinate collaborations with other epidemiologists who work with the Wisconsin Sleep Cohort. And she's her research interests include understanding the physical and social influence on sleep and health across the life course. So, and then she'll be followed by Matthew Gratton, who's a sleep researcher and doctoral student in clinical psychology at the University of Kansas. And he's researching, sort of trying to leverage informatic techniques on helping to characterize and treat behavioral sleep disorders. So thank you so much for joining us today. We'll turn it over to Dr. Hagen.
Oh, sorry. There we go. Okay. Well, thanks so much for having me. I'm just going to briefly talk about the Wisconsin Sleep Cohort study. I'm going to share a little bit about some key findings. I'll then share about the data that's available from our cohort on the NSRR website and talk about some possible uses of our data, and then describe our experience with sharing, and we'll have our an example of a collaboration that came out of this shared data. So just briefly, the Wisconsin Sleep Cohort study is an ongoing longitudinal study of the causes, consequences, and natural history of sleep disorders, and particularly sleep apnea. We use overnight in-laboratory sleep studies that were conducted with a baseline sample of over 1,500 Wisconsin state employees assessed at approximate 4-year intervals. Additionally, there are several mailed surveys that have been completed at various time points over the past 3 decades that have collected myriad self-reported data. We have had ongoing and overlapping NIH funding since 1988, and though all these protocols have included various data collection that has come in and out of our protocols. So our the data that we have are sort of a complicated mishmash across that time, but it creates a pretty rich data set that we have on the cohort of study participants. So I'm just going to do a real brief whirlwind of some key findings. I'm not going to highlight everything, but there's just a few things that I'll talk about just to give a sense of the breadth of what the sleep cohort has published over the past 35 years. So the initial prevalence paper, which is what sort of put the sleep cohort study on the map, came out in 1993. The importance of this paper was that it really showed that obstructive sleep apnea was not rare, and that it affected many people. So there was a 9% prevalence among men and 4% prevalence among women 30 to 60 years of age. And then in 2013, we updated these prevalence estimates using the additional years of data that the sleep cohort study had collected, and then used national age and BMI information to estimate what the national prevalence was based on the findings from the Wisconsin cohort. So the updated prevalences from 2013 were 10% in men 30 to 49 years old, and 17% in men 50 to 70 years old, 3% in women, and 9% in women who are 50 to 70. So this is just evidence of increasing prevalence that mirrors increasing obesity trends in the country, as well as an aging population. So a lot of what the sleep cohort study focused on in the first half of the past 35 years was obstructive sleep apnea. We have papers showing that OSA is associated with higher risk for incident hypertension, incident nighttime BP non-dipping, incident depression and stroke, as well as all-cause and CVD mortality. So after, while we still look into various aspects of risk for and consequences of sleep apnea, we've also shifted our focus over the past 10 to 15 years to sleep duration, sleep quality, as one of our, more of our major focus. So there've been a lot of work in the previous year or the more recent years about sleep duration. We found that short sleep duration is associated with metabolic hormone dysfunction. Less restorative sleep over time is longitudinally associated with higher BMI. We did a paper where we found that sleep duration increases and sleep timing changes with retirement, which might feel like something that you might expect. But we were able to document that there are substantial changes that happen in people's sleep with the role change or the life event of retiring. Subjective sleepiness, we find is associated with a higher risk of depression, and midlife insomnia is associated with earlier retirement. So that's just a smattering of work that we've done with our data in recent years. And we are thrilled to be able to share our data through the NSRR platform. There's more work than the staff scientists of this study can do on our own, and being able to share this with the wider sleep community is a great way for work from all this data that were collected to yield more important information in the sleep field. So we have shared a lot of the data that we've collected. That includes polysomnography variables, body habitus, blood pressure, and some other cardiovascular data, self-reported health habits, including smoking and drinking, physical activity. We have information on anxiety and depression, mortality data, self-reported sleep duration, sleep quality, and sleepiness data. We have data on medications, and then we also have more data that hasn't yet been shared. We're currently in the process of working on sharing some of our our napping data. And there's additional data that's we're using currently that, once our current grant cycle, once those wrap up and we write a few papers ourselves, we'll be sharing even more data that we have. So there's a lot that can be done with the data that we have shared, and there's a lot that I'm sure no one in our group has currently thought of, which is wonderful. But there is a lot that we know of that can be done with the data that we have. So our PSG data has been used by lots of people for machine learning, but it can continue to be an excellent source for that kind of work. Those models will be refined, and the PSG data that we have will be useful for that, from now on. We also have information about circadian preference. So somebody could look into the longitudinal association between circadian preference and PSG parameters. We have sleepiness data. So we currently have subjective sleepiness data that's shared. Some objective sleepiness data is available, and we're in the process of being able to share additional objective sleepiness data. So there's a lot that could be done to look at associations between multiple dimensions of sleepiness and variable ways of characterizing sleepiness, and those associations with many outcomes and predictors. Like I said, we have napping information, and there's a lot that could be done with looking at naps longitudinally and napping in association with lots of outcomes and additionally predictors. And of course, much more that we haven't even thought of. We have had a very positive experience working with the NSRR staff to go through all the logistics and nuts and bolts involved with sharing our data. We have a pretty complicated consent history because we started in 1988 when sharing data wasn't the same as it is today. And we have consent forms that were created every approximately 4 to 5 years that changed with the different protocols and were shaped, by the norms of the day. So we have had, a rather complicated process working with our IRB to figure out which of the data that we can share. And once we did figure that out, we've had a really smooth time working with NSRR to logistically make that available. The model that we use with our study, in part because we still have, an ongoing, we're actively ongoing, we're currently collecting data, and we have plans to continue doing so. So we have investigators that are actively using our data that are part of our team. And in part for that reason, and in part because our data set's rather complicated and our consenting history is pretty complicated, we still have oversight over who we share data with. So the NSRR staff screens things first, screens requests first, but then they get passed along to us. We have about, I think, an average of 3 to 5 requests per week. They get reviewed by myself and Dr. Paul Pard, who's the overall PI and director of the Wisconsin Sleep Cohort study right now. So we look through them and we approve them. We've approved over 200 uses of our data, I think over in the past about a year. And we have users all over the country and around the world. And similar to what Susan was saying, we have everyone from high school students to tenured professors requesting and using our data. And now I would like to introduce Dr. Gratton, who's going to describe his, he is somebody who has used our data and it's resulted in some great work.
So I just want to make an amendum to that. I am not a doctor yet.
Sorry about that. Future Dr. Matthew Gratton.
Yeah, presumptuous, but yes. Just share my screen. All right. So, hey, everyone. Thanks for inviting me to talk about my experience with the NSRR. I'm a graduate student in the clinical psychology doctoral program at the University of Kansas and University of Kansas Medical Center with doctors Nancy Hamilton and Diego Mazzacci, respectively. I research physiological and somatic relationships of sleep behavior using informatics techniques. This approach to research requires access to large data sets. So as such, I have a lot of experience with big data. A recent example of that being my involvement in the creation of a database of sleep studies at the University of Kansas Medical Center with Dr. Mazzacci. So just want to kind of go through my story with the Wisconsin Sleep Cohort. After receiving encouragement from Dr. Mazzacci to explore the NSRR for any studies with variables that fit my research interests, I found the Wisconsin Sleep Cohort. The Wisconsin Sleep Cohort was attractive to me, specifically due to its inclusion of a self-report nightmare variable and behavioral metrics, along with physiological sleep measurements. The Wisconsin Sleep Cohort is sort of unique in this inclusion within the NSRR and other publicly available research databases. And after my poster was accepted to the meeting of Associated Professional Sleep Societies, I reached out to Dr. Paul Pard and his team, which includes Dr. Erika Hagen and Dr. Emmanuel Mignot, to discuss access to the Wisconsin Sleep Cohort and stages data that was previously available on the NSRR. So after a quick discussion, we decided that we wanted to connect at Sleep and discuss future access to the data. Not only was I able to get access to the requested data, but this discussion also prompted the future dissemination of the MSLT NAP data to the NSRR. And here's a picture of me presenting my poster at the APSS this year, photo courtesy of Dr. Mizacci. So yeah, just to give a high level understanding of the results, this is a representation of the cross-sectional data that we examined. We looked at depression, anxiety, and nightmares in 758 participants from the Wisconsin Sleep Cohort. We examined higher frequency of nightmares represented on the x-axis. And it was associated with higher levels of anxiety symptoms, which you can see on the left plot, and depression symptoms shown on the right plot. That led us to question whether depression and anxiety symptoms would be associated with increased incidence of higher frequency of nightmares. And we're currently exploring the longitudinal data to test this hypothesis. So, in conclusion, my access to the Wisconsin Sleep Cohort through the National Sleep Research Resource opened a door to a current publication, and that it led to a data set that I'm now using for my thesis. Using my personal experience as an example of the power of the availability of the Wisconsin Sleep Cohort, I think there are a couple of strong takeaways. Firstly, junior colleagues may be in an important but overlooked role that may be uniquely suited to the immediately available nature of the NSRR data. Junior researchers such as fellows, residents, and even undergraduates don't have the time or resources available to collect a large sample of data that they need in order to complete their milestones in a meaningful way. And obviously, while there are limitations to using a data set past its intended research question, there's still copious data that can be mined for the use of furthering our field. Of course, it's important to acknowledge those people and entities that helped me get to this point, as well as funding sources. Sincere thanks to the NSRR as well as the Wisconsin Sleep Cohort study for affording me the opportunity to perform this research and speak here. And thanks to my advisors, Dr. Mizacci and Nancy Hamilton. If you'd like to reach out, my contact info is on the right, #SleepPeeps. Yeah, thanks very much.
Fantastic. Thank you so much. As I don't see any questions right now, let's move on to the next talk. So this was a really nice example of, how these research cohorts can come into the NSRR. But now we'll talk about how this works from a clinical perspective. And so next up, we're going to have Dr. Harlin Lee talking about Nationwide Children's Hospital Sleep DataBank. And Dr. Lee is an assistant adjunct professor at UCLA Mathematics. And her research is really using things like graph theory and subspace reduction and sparsity metrics to look at ways of approaching healthcare and social science. So she'll be talking about both her data set that she's working with as well as collaborations and scientific results.
All right, thank you so much. Can you see my screen okay?
Yeah.
That's great.
Okay, great. Thank you so much. So good morning from LA. Thank you for the very kind introduction and invitation to talk here today. I'm Harlin, and I am very excited to talk about NCH Sleep DataBank, including a little bit about the process behind the scenes of how it got to NSRR. So if you go on the NSRR website and look at the list of data sets, you can see the NSH Sleep DataBank right there. And first, let me talk briefly about the data set itself, focusing on the characteristics that makes it rather unique. So first of all, as you may have noticed from the previous slide, this data set is enormous. It's truly big data. There are in total more than 3,900 sleep studies or PSGs collected over 2 years from 3,600 unique patients. And each of these overnight sleep study contains dozens of different modalities, as well as the sleep technicians' annotations and sleep stage classifications. So we have signals like EEG, ECG, etc., and snoring, airflow, But we also have comments about patient movement or sleep stages. So you can see an example of an annotation right here. For example, 16,000 seconds after the start of the sleep study, the patient moved their limb. And we have both of the record by human sleep technician and then also by the software. So with all of these files and also the derived variables by the NSRR team, the entire data set is at 2 terabytes at this point. And to the best of my knowledge, this is the largest curation of pediatric clinical sleep studies to date. Secondly, it explicitly focuses on children. So the medical data that you see here are collected from Nationwide Children's Hospital in Columbus, Ohio, hence the title NCH, which is one of the largest pediatric healthcare systems in the US. On the right, I have the age distribution figure of the patients in the data set, and you can see that majority of the patients were under 15 or even 10 years old at the time of sleep study. And 20 patients who were more than 30 years old are just not shown in this figure. Next, the data is not from a controlled clinical trial environment, but it's everything that was done as part of standard care at NCH, the hospital. So this screenshot is an example of what a clinician or a technician might see at NCH Sleep Laboratory during a sleep study. Each of these lines indicates some physiological signal that's updating real time, as well as these annotations that comes from both the sleep software and the technician if they have any comments. And we just exported all of these signals and all of these comments. So on one side, this means that the data is obviously very, very noisy. But on the flip side, it also means that it is a close reflection of truly the real world clinical setting. And I believe that can be very valuable, especially for data mining or data-driven computational research that depends heavily on the available data set. Lastly, in addition to the sleep study files that I just talked about, we also published the electronic health records data, the EHR data. There are accompanying EHR data pulled from the entirety of patient history at NCH, not just between 2017 and 2019 when we collected the sleep study data. So what that means is if a patient had at least one sleep study between 2017 and 2019, we pulled that patient's every single record in terms of demographic, diagnosis, medications and procedures, anything that was available in the software that was used at NCH at the time, from their very first visit to NCH. So all of this totals to about 5.6 million rows in the tables, which are available for download at NSRR. Oh, I don't know why that didn't load. Okay, that's fine. Going beyond just the sheer volume of EHR data, this figure is not showing for whatever reason, but it's supposed to show you that we have every patient's medical history going back years before their first sleep study in the data set, as well as their follow-up for a couple hundreds of days. So this information could allow researchers to do retrospective studies on patients, which I think is a very exciting research direction that can use this data set. Now that I've given a quick overview of the data set, I'd like to discuss kind of the behind the scenes of how this came about to NSRR and what that process has been like. So in 2017, Dr. Linwood and Dr. Chi got a collaborative NIH grant to collect data and also develop mathematical methods for multimodal physiological monitoring. Since then, Dr. Linwood moved from NCH to UC Riverside, and Dr. Shi moved from The Ohio State University to Carnegie Mellon University, which is where I did my PhD. And Simon, who's a medical doctor, was the chief information officer at NCH at the time. And he had already named NSRR as one of the potential data depositories and collaborators in the grant proposal. Then for the next 2 years, the sleep studies were collected at NCH. And just to reiterate, everything was done as part of their standard care, following their usual protocols with their usual patients. Afterwards, I joined this project as UHS PhD student at Carnegie Mellon University. You can see me as a young kid over there. And at that point, the team at NCH was working on exporting all the tables, the signals, doing quality check and de-identification, which we all know is very important for clinical data. Then I afterwards took charge of performing data verification, doing the preliminary data analysis, and prepared the dataset manuscript as well as Python code that can interact with the dataset to be released open source. Then we shipped this large dataset to NSRR on an encrypted hard drive. Because since the dataset was so large, there was just no way we could transfer them online at like a reasonable speed. Well, we all know what happened in 2020, so there were some delays everywhere. And it took a couple of months for the data use agreement between NSRR and NCH to be fully executed. But once that was done and the COVID delays were over, the dataset went live in 2021, February. And this is a screenshot of what you would see if you click on the dataset. And then finally, our dataset paper was published this year at Nature Scientific Data. And I want to make a note here that during the peer review process, we realized that we had to make an update to the dataset. And the NSRR team responded very quickly. They immediately put the data request on pause while we from Carnegie Mellon and NCH were figuring out what changes to make. And once we made the edit on our end, NSRR updated the dataset right away and resumed download. So there was minimal disruption in terms of the science. So working with NSRR was a very easy process, and the team are super responsive. Also, for this data set, NSRR has full control over approving the data requests, which was a perfect fit for our project because the NIH grant has expired and we don't have a permanent staff for governing and sharing this data set. I want to conclude this talk by showing a quick example of a follow-up project that I've done with this data set. So this is for a problem called sleep scoring or sleep stage classification, which is a problem where given 30 seconds of PSG or sleep study signal, you're supposed to give a label as one of the 5 sleep stages: awake or rapid eye movement, REM, and non-REM stages 1, 2, 3. This is currently done manually in a lot of places, which I'm sure you can imagine is extremely time-consuming and costly because you need to do this for every 30 seconds of sleep when people sleep 8 hours a day. While there has been a lot of effort to automate this process using both rule-based and data-driven approaches, this is very much an unsolved problem for infants, children, and adolescents, which is why NCH actually does this manually. So the sleep stages you see in our data set are done by the technician and approved by physician who is board certified in sleep medicine. And pediatric sleep scoring is very under-researched compared to adult sleep scoring in the context of machine learning for health and well-being, partially due to lack of relevant data so far. So me and my collaborator thought, okay, a large-scale pediatric multimodal dataset like NCH Sleep DataBank might be a really good fit to try out a machine learning approach to this problem. So with my collaborator, Akib Saeed, who actually reached out to me by finding this data set at NSRR, we trained a neural network model, something called Transformer, on 7 channels of EEG signals. So the details of the architecture is not important at all. Just on the high level, this particular architecture learns representations of time series signals with a focus on what part of the signal is important when, which is why it has seen a lot of success in speech processing, image processing, and natural language processing, like something you would see on Google Translate. Then once the transformer model learned features from the multi-channel EEG signals, we trained a very simple classifier on top of that to predict the sleep stages. Overall, it got a 78.2% accuracy, and you can see a more detailed breakdown on the right with this confusion matrix. So you can see that, so you can see on the diagonal side that 75% of the wake signals were correctly classified, 50% on N1, and so on. Another way to kind of verify our results is to pick a random sleep study and plot their hypnogram, which is what you see, to show how their sleep stages changed throughout. So on the top row, you can see the labels that were given by NCH in the data set. And then, the patient starts being awake and then moves through the non-REM stages and then wakes up again and so on. The bottom row is prediction by our machine learning model. You can see that some parts are close match, like here, and some parts are not so much. So there is certainly a lot of room for improvement. We have put up our current results as a preprint, which can be found in this link, but we believe that there's still a lot of work to be done here. And I think that's pretty exciting. So to conclude, this data set at NSRR it's a pretty exciting pediatric clinical studies data set. And I think there are many ways to be used for further research. So some potential use cases include automatic sleep scoring, like I mentioned, especially something that can combine PSG modalities beyond EEG and ECG, which I don't think has been really explored in current literature due to lack of large data. Another thing that I think will be really exciting is real-time sleep disorder alert. So as so something that can detect symptoms at their onset, which can be really useful for something like obstructive apnea, and that can be really critical for small children and infants. And such things like diagnosis prediction, identifying patient subgroups, and treatment efficacy analysis. The latter 3 would be combining EHR data with the PSG data. So that's all I have for you today. Here's my website if you want to get in touch with me, and thank you so much.
Great. Thank you, Dr. Lee. That's really wonderful. And I just wanted to add that both of these data sets came into NSRR as exceptionally high quality, high value data sets. And on top of that, what's really exciting is that with the tools that we have with the NSRR, we can in broad scale enhance them by adding things like harmonization, alignment of different epochs, and all of those other little nitty-gritty things required to make sure that everything is right in order. So it was very helpful to have such great data sets coming in, which really facilitate the NSRR's ability to truly take these to the next level in their ability to interconnect with other existing data sets and thus expand the scope of the scientific research performed. So thank you very much. Again, we have no open questions right now, so we will continue on to our last talk. And I am very, very pleased to introduce my exceptional colleague, Paige Sparks, who's been wonderful to work with. She's a clinical research manager at the NSRR, and she plays a key role working with new data ingestion. And also we've worked together on outreach and engagement for the NSRR. So really happy to have you speak, Paige.
Thanks, Mike. So I'm going to give a high-level overview of how to share data with the NSRR. And if you have specific questions, I'm happy to answer those. So you can use the Q&A function. I am also grateful to our speakers who have covered some of these questions in their own talks. So why share data with the NSRR? I think this quote from the NIH and their recent data sharing policy really captures the essence of that. But the main focus is from the NSRR's perspective is to advance robust scientific discovery. The harmonization efforts from the NSRR team help maximize impact of those data sets. You can meet or fulfill journal or funding source requirements. Many journals and soon the NIH will require that your data is shareable. And the NSRR provides investigators who contribute data with built-in infrastructure for data sharing. So as was previously mentioned, once the grant has ended, there's still a team here to manage your data and all of the aspects of data sharing. I'd like to highlight the new NIH data sharing policy, which will be effective January 25th of next year, 2023. I've linked some of the relevant links here on this slide, and I'll pop them into the chat after the talk. The policy, they also have a guide for selecting your data repository. And of course, the NSRR meets those desirable characteristics for a data repository. And they also have a list for domain-specific repositories. And if you search for sleep, the NSRR obviously comes up. The NIH is going to require researchers to plan in advance how they are going to manage and share their data. And I encourage you to register for our third webinar, which will be held in November, where we are going to highlight the things that our team is doing to help investigators prepare for this change and make sure that, as a few of our speakers have mentioned, you're planning where it is that you're going to deposit your data and how it will be shared. So in terms of the steps for sharing data through the NSRR, I think about it in 3 different stages, so to speak. We initiate, transfer, and then share out the data. For the initiation, you can contact the NSRR. The link is in the chat at sleepdata.org. We ask you to fill out a share form, which gives us information about you, about the project and the data that you have available. And then we reach back out to you. We'll have a meeting. You can meet with our team members, Dr. Perrow and I, occasionally Dr. Redline or Dr. Purcell, depending on the project. And we walk you through this entire process, answer questions. If you have specific concerns about data sharing or specific hurdles, our team will walk you through all of those. And then we initiate the regulatory approvals. So that might be an ethics committee or IRB approval, as well as data use agreements for sharing the data itself. For the transfer, we ask that you send data dictionaries, of course, the finalized data sets, and then any documentation that might accompany your data set to our team. So manuals of procedures, annotated data collection forms, things along those lines. And then we put all of that information onto a sharing server or a staging server so that you can review exactly how your data is going to look before it's made public. And then once you've approved it, we will publicize your newly available data through webinars like this, our newsletter, the website, social media, etc. A little bit more in detail, what you are going to be responsible for as someone who is contributing their data is to complete that initial share form, provide the NSRR with information that we might need, like a brief summary of your data, consent forms, or things along those lines for IRB approval. All the documentation, such as those annotated questionnaires and manuals. The final data itself, as well as data dictionaries. You'll be responsible for removing identifiers from data sets and the raw data files, and our team can assist you with that. Responding to any queries that we may have as we're preparing the data for posting, if we're curious about how specific variables are defined or something along those lines, responding to that is helpful for us to document your data best. And then, of course, to securely upload all of your files to the NSRR. Our team is here to assist you with each step of the submission process. So if you have questions or concerns, we are here as you are working to deposit your data. We will establish an institutional data use agreement. We now have a uniform template that we use for all of our data cohorts that includes information about how your data will be shared. We also make sure to review all the data that we've received to make sure that there are no identifiers and that participant or patient privacy is maintained. And then we create a repository for your data set to organize all of the information, the data, as well as the documentation, which you've seen in some of the talks today. The data harmonization, I think, is something that really sets the NSRR apart. It maximizes the usability of your data, especially in a big data context. And we'll talk more about that in future webinars as well. As mentioned, we make sure that folks who access your data are only the folks that you want to access your data. For example, if your data is not to be used for commercial purposes, we ensure that that happens. Or if there are any other restrictions, such as topic or domain-specific restrictions, we handle all of that. And then we publicize your data set, as mentioned, through the websites, social media, and newsletters, and provide you some updates with the use of what's happening with your data through the NSRR. Additionally, as Harlin mentioned, I think sometimes there are user questions or feedback, and we can also help you to facilitate that interaction between users who are using your data and your team or the original collectors. If there are any questions, I encourage you to reach out to support@sleepdata.org. Our team is always happy to answer questions or point you in the right direction if we're unable to. Please follow us on Twitter, and if you have not yet joined, I encourage you to join sleepdata.org. Thank you very much.
Thank you so much. And with that, I think we have been able to successfully have our first webinar. And I just want to thank all of our speakers for joining us today, and all of you for joining us as well. And please make sure, if you can, come to our next webinar. We're going to be talking in detail about fair and trust principles. We're going to be talking in detail about data and signal harmonization and some of the challenges and goals that we have with the NSRR at our next one. So I think this is going to be really useful, especially in planning ahead. If you plan to be collecting data that will be shared, thinking ahead and really helping to facilitate taking that to the next level in terms of what's going to happen. So I really appreciate everyone's time today and look forward to seeing all of you on October 12th. Thanks so much.
Thanks. as people are just signing off too, I just encourage you to reach out to our support line or individual emails with